Hello and welcome to episode 4 of the Papercast with me, Dominic Hill. My guest today is Ryan McClory, and we are going to be talking about his paper in the journal Forest Ecology and Management called Spring Phonology Dominates Over Light Availability in Affecting Seedling Performance and Plant Attack During the Growing Season.
Catchy title.
It's very good. Yeah, very good. At least I haven't got any puns in it.
Yes.
My supervisor, sorry Luke, is absolutely obsessed with puns in titles, and he works on rocket. So everything is rocket science.
Because my undergrad thesis was follow your nose, like the effect of volatiles on Helix Aspirus or something like this. So
yeah, people love a pun title. If I was a reviewer, I'd get rid of all of them. That would be all I would say. Like, everything's fine. No puns.
Yeah, absolutely.
So I thought we should begin because I've in the last few episodes I've been kind of jumping ahead of myself a little bit. So I want to take it back, set the scene a little bit, and talk about your PhD project as a whole. So could you tell us a little bit about your PhD?
Certainly can.
Generally?
So my PhD is basically on oak masting. So oak trees will produce acorn crops once every 4 to 7 years. It's not exactly that, but basically this is what people tend to say. And they'll do this in sort of a huge boom of acorns in 1 year and then not very few in the preceding and years after. They think this is partly maybe just to sort of starve seed predators and stuff like this. So when you do have a mast year of all the acorns being produced, there's not as many predators about, and those that are about are sort of going to be satiated quite quickly. But I'm looking at sort of the mechanistic drivers of what causes this mast. So it's important to know this because we've got, especially with the COP and this, the government wants to plant so many trees. I think it's something like 30,000 hectares by 2025, 2024, somewhere around there. And they need for this to know where the seed crop is coming from. And species like oak, where they do this masting behaviour, as well as having acorns that are recalcitrant, meaning they dry out very easily and you can't store them, you need to find a way to get consistent seed crop to keep up with the demand of planting forests. So ideally, by the end of my PhD, I'll be able to suggest basically ways in which we could either improve seed crop to keep it more consistent, or I could have something that could sort of pre-warn us as to when a mast year or sort of certain areas are going to mast. Yes, that's basically it, I think.
Fantastic. And how long have you been doing the project so far?
So I've been here for about a year and a couple of months now.
Okay, great.
Like this.
Settling in?
Yes, I think so. I started sort of mid-COVID, as I'm sure a lot of people have this story, so there was no one here. And then you have to meet everyone over Zoom and Teams. It's not so conducive to sort of socialising because, you can have sort of one-on-one conversations, but as soon as there's a group of, more than 10, suddenly you can't have conversations between, so one person has to talk at the same time. But I feel now I'm getting a bit more into it and sort of used to it, and especially now everything's starting to open up a little bit more. We can do this, for instance.
And this is great. It has been difficult, and people might not know exactly what it's been like to be a PhD student during COVID. And you're right that it's been difficult, especially that sort of collaborative element that PhDs draw upon so much. Normally you're in the cafe or whatever at lunchtime, someone will say something and you go, oh, that's a great idea for what I want to do. And we have missed out on a lot of that, and it's a real shame, but it feels like it's getting back, because I started just before COVID, like 6 months before. And then now it's starting to feel still not totally back to normal, but it is starting to feel more similar. What was your experience of the PhD during the sort of extreme COVID lockdown times? What were you getting up to?
So I was fairly fortunate where I could we'd already decided where I was going to do my main study, which was in White and Woods in Oxford. So there you had the ability, even during lockdown, to go out there and start some data collection, just because you were in the middle of the woods, no one around, stuff like this. You had to take certain measures, like, I wouldn't have been able to pick someone up in the car, obviously, and go and do field work, but it was me and my girlfriend that did sort of any initial field work. So I was fairly lucky there. It meant I could get stuff done, and then the other part of the PhD, the more collaboration, meeting people, could sort of be put on hold. Obviously I didn't do any sort of attempts at, talking to people and meeting up, but yeah, all over email and Zoom and stuff.
Yeah, it's just not the same, is it? But it's good that you did manage to do some field work during COVID, because I mean, I can do anything. All my work is at CEL, the crop lab, and obviously you just weren't allowed in.
I always think the worst time for COVID to have hit would have been like, basically 4 months from now, almost. You're sort of just in your second year, when you've got your stuff ready, and then you can start the experiments. Because I think very at the beginning, I think people were overly cautious, as they probably should have been, and just basically stopped everything.
Yeah.
But that meant certain stuff like me wandering around the woods probably wouldn't have been allowed even then. But because I think they did shut them for a bit. But and then when they realised it was like, well, actually, you're in the middle of the woods, no one else around, it's sort of okay to do.
And oak trees can't get COVID.
Oak trees can't get COVID.
Okay, excellent. That's good.
I don't think squirrels can either, but
Do we know that for sure, though?
Well, there's been no study, but you've got to, and some things you've just got to accept.
So how long do you have left? Are you a 3-year or 4-year?
3.5 years funded, and then I'm allowed to go to 4 years, aren't I? But I have to ask very politely for a bit more money, I think.
Yes, I imagine you probably will do. But with the COVID thing, that is quite easy. I just got approved for, I think, 3 months extension,
nice.
Which is, great, but I lost almost a year.
Yeah. Why do you have a weekend?
I mean, I managed to publish my review, and that's all I did. Yeah, and I'm quite pleased with that, because it was I had the time to put into it rather than doing it. The guy I was speaking to, Eric, in the last episode, is writing his literature review now, and he's nearly ready to submit. So it's going to be. I mean, I'm sure he'll do a great job, but it is going to be more rushed. Whereas I had nothing else to do. Great. And what's your experience of the project been like so far? Are you enjoying it? Is it stressful?
Yeah, I'd say PhDs seem to be stressful, partly because everyone tells you they're very stressful.
Definitely.
So I think you can never have the opportunity to relax, because then you're like, well, why am I relaxed? Because people said it should be really stressful. So you can never sort of settle. I mean, it very much sort of comes in waves, like grief. And it's just... so you'll have a period of being like, oh, great, I sort of feel like I know what I'm doing. And then the next week something wouldn't have worked. And then you're like, well, maybe I should just leave and go live in the woods or something like this. But no, I'd say generally I feel the project at the moment is still going well. Part of being sort of locked in and not being able to meet people is, I suppose, a lot of my time, has been spent trying to work out where to get data sources from and, try and do collaborations over email. So I've been focusing quite a lot on that, so it feels like it's worked reasonably well. Yes, pretty much.
Yeah, I think your point about the stress levels of it is so on it. It's so accurate. You do feel like every time you feel like things are going well, you feel like they shouldn't be.
Yes.
And I think a big part of doing a PhD is learning to be okay with the sort of ups and downs of it. Yeah. Because it, like you say, an experiment can just go wrong, and there's nothing you can do about it. Yeah. Like at the moment, my potato plants have blight. I can do nothing about that at this point. Yeah. There's things I can learn from it and try and avoid it next time. But you just have to be like, okay, well, this is what's happened. Yeah. Can I make the situation better? Can I change the things I can change, and can I accept the things I can't? And how can I try and get something from it? Yeah. And I think people struggle with that, obviously, because it's not really how you go through the rest of university. You don't really do a lot of independent work. You have to learn the answers to the tests, and if you learn the answers to the test, you do well. Yes. And then you come here, and it's like, well, there's all these forces conspiring to try and...
I mean, especially with any sort of ecology, natural world stuff, it does get, you can plan so much, and then, the weather will just fuck you up for the whole year. And then you're like, oh, I guess. Yeah. I mean, I was always told a story of some PhD. She wanted to go and study hummingbirds in Brazil or something like this. So she'd set up, done all the organisation. It was going to be here for 3 weeks and stuff like that. She'd have to do all of her work then, all the data collection, no other money to do anything else. And she turned up and it rained for the whole 3 weeks. And apparently hummingbirds don't go out in the rain. So she just had to be like, well, I'm in a rainy Brazil for 3 weeks with nothing to do.
You literally just have to be like, this is the situation. Can I do anything? There's a reasonably well-known story that Liam from CEL likes to talk about, where a guy literally just put all his eggs in this one experiment. And not literally, metaphorically.
Study eggs.
And the crop just failed. It didn't rain or something, and the whole crop that he was supposed to collect all of his data from, because everything before that had just been toying and fiddling to try and get this one experiment perfect. So it was like, trying to figure out which soil to grow it on. It wasn't like a big experiment. It was just like, how well do these plants grow? And then got everything exactly how they wanted it, perfect. Planned it to within an inch of its life, and then it just didn't work. And you just have to crack on. There's literally all you can do. You can cry for a bit, but and my supervisor gets a couple of emails every month of me being like, I'm done. And then by the following week, I'm like, I'm fine. I'm fine. We'll get through it. And how's it been? Are you where you thought you would be? I guess you're probably not because of COVID.
Well, it depends, because I'd started this as well knowing that COVID was a thing. So I sort of had ideas of what it would be like a bit. It depends sort of when you're asking what I thought it was going to be. So at the very beginning, before I'd even actually been accepted to the PhD, I sort of had these vague ideas of like, I bet there's some long-term record on acorn crops and stuff like this, so I can sort of turn up and then like my own field work. Yes, that would be stressful getting together, but at least I'll have this big data set. And then it was like the first week in where I was told that basically, no, there was no large. You've got to find the large data set, Ryan. We don't know where it is. I was like, oh, okay then. So, but then from that point, I'd say it's all gone more or less to plan. I have these sort of vague ideas. It's very... you can make really good plans, but you've got to sort of just go with it in a lot of different directions. So I think I've got a good amount of data in different areas now. So I'm probably about where I want it to be. So yes, more or less, but we'll see. It could all still go wrong.
It could. It could. That's something you always have to bear in mind. I guess a lot of people think that there are these data sets out there, these mythical data sets, and there just aren't.
No.
There's great data for some things. Like if you want to know what the weather was 30 years ago in a square mile.
There's some guy that's been, they've been recording the temperature in a field for the last 200 years.
But anything useful. There is no data. You have to find it yourself, which I think is probably a good thing, because it trains us to be able to go out there and find out what we need to know.
Yes.
So with these papercasts, I like to try and follow the structure of the papers, because I think that is logical and it makes sense. For people that aren't aware, generally scientific papers will go abstract, which is like a paragraph that just tells you everything you need to know, really. I mean, if you just read the abstract, you'll get a pretty good sense of what went on. You won't get the nuance, and you won't necessarily be able to think critically about the paper, but you'll get the gist. Then you have the introduction, which sort of sets the scene, and then I don't know why I'm looking at this. I know how it works. Then you have your materials and methods, which tell you, or should tell you, exactly what happened. And in theory, you should be able to look at it and go, I could do that again. I could repeat that even without talking to the person that published the study. In theory. That isn't always how it is, but that is how it's supposed to go. Sorry about that. After your materials and methods, you have results, which is exactly what it does, what it says on the tin, as it were. It's just everything you found in a synthesized way. It's the data analysed normally, and often you'll have the raw data somewhere in the supplementary materials. And then at the end, you just have a discussion where you talk about the results. And the results, you just say, this is what we found. You don't elaborate at all. You don't say, well, we think it's because of this or that. That's all for the discussion. So I'm sure 90% of people watching this
that's good. I've
know
learned
that.
That's good. I'll do my next
Thank you.
one.
I try my best. I really do. So we'll start with the introduction, and this is often a difficult question because it requires you to remember quite a lot about what you wrote when you wrote it, which
perfect.
is when did you say you wrote it?
So the work happened in 2018.
Yeah.
And then I sort of wrote it. Yeah. The main of it, obviously, because it's from my thesis, would have been sort of 2018, 2019. And then you go back to it and you just edit bits for a long time, basically.
Yeah. And then did you submit it after you started here or before?
Before. So this is part of the reason it was done so long ago is my supervisor at the time was essentially like, he thinks it's quite a good paper. We should probably aim high. So we aimed high first, and we went to sort of very high impact journals. And then it was a series of rejections of different...
They're fun, aren't they?
Yeah, well,
really
the first one, I was like, oh, if this is what rejections are like, that's not too bad. Because basically one reviewer was like, yeah, sure. The other one had some qualms, and then the editor eventually sided with the one with the qualms. But yeah, I think one of mine, they were basically like, why are you bothering to try and live? Like, just stop whatever you're doing, you know?
Wow.
How dare you? Yeah,
Wow.
yeah. But interestingly enough, there the other review as well was then like, oh yeah, great paper, fantastic.
Yeah,
so.
it does always seem to go like that. You have like, I don't know if they pick them. They have some way of picking them, but it does seem to be a good cop, bad cop kind of thing.
Yeah.
When I did my literature review, one of them, it was like he was another supervisor, or they were another supervisor. I think it was a man, because on Frontiers they tell you who it is at the end. But it was literally like, I think these things would make your paper better, and then I did it, and the paper was better, and he was like, good job. Yeah. Like I think he literally at one point said, good job.
Yeah.
And then the other one was just like, this is horrible.
Yeah.
Like I've talked about this before, but I had lots of old references in my paper, or in my review, and I said in the introduction, I have lots of old papers because all the evidence on this topic is old. There is no new evidence. Yeah. And that was like the last sentence of my introduction. Like, here's what we know, but it is outdated, and that's sort of the point of doing this, so that I can start updating it. And the reviewer was just like, where's all the new... I was like, there is none. I told you there is none.
This is the study.
This is literally it. But that is just how it seems to go. And peer review is a great idea. But in practice, it can be extremely frustrating.
Yes. Yeah.
But you got this one published in the end.
Yep.
So we'll crack on with the introduction. Is there any sort of important previous research, or maybe not previous research, but information? I mean, you've already talked about masking a little bit, but can you just help people get into this paper a little bit and understand what you're going to talk about?
Yeah. So with this paper, it's not really linked to masting. This is, my PhD is about masting, but this at the time was just a master in ecology and biodiversity. So I had to choose something in the area where, I could sort of research and do a study on. So this one I had interest in sort of phonology and phonology of plants and how, different phonologies and variation because of climate change or just because of the sort of spread of a species phonology generally can affect their survival or performance. So this was sort of based off an interest of mine where it was, I like researching how phonology can change ecosystems. So this was sort of the level of study I could do at my master's level. And yeah, it seemed to come out quite well in the end. But yeah, it was sort of that sort of area. There wasn't one paper that made me do it. It was just a general interest in phonology.
Yeah, well, I guess for your masters, that is sort of more how it goes. You can't really get too deep into the literature, whereas with a PhD, you get a lot more time to do that sort of thing. Can you tell the people what you mean by phonology? Because that is a bit of jargon.
Fair. So my girlfriend's normally the one that says that. She's always like, what the fuck are you talking about? So yeah, phonology is basically the timing of biological events. So when leaves come out in the spring, that's spring bud burst, that'll be a sort of phonological event. And it's not an issue as such, but basically with climate change, some of these phonological events are sort of led by temperature or sunlight and stuff like this. So they can be changed and altered with climate change. So a very popular sort of area of study is phonological mismatch, where you have the best example, sort of birds that migrate for winter. So they have sort of timed their migration to be there ready for when the leaves start growing and fruit start appearing on trees. The issue is if climate changes faster in their area, where the birds initially are and are going to go from, it could be that they start to migrate earlier and then there's no leaves or berries or anything when they get to their end point. So this is what I was doing with mine, was more just trying to understand how seedling germination, so when they first germinate from the acorn, how that could change and interact with light availability, and then whether that had any effect on sort of plant performance, attacked by herbivores or sort of any pathogens.
Great. Yeah, I think there's a few projects at Reading that obviously unrelated to yours, but that look at this phenologic mismatch idea. Is it phenologic mismatch?
Phenological mismatch.
Phenological mismatch.
Yeah.
There was, I don't know if they finished now, but there was someone that was looking at the timing of flowering in some crop and pollinator availability, because it was, it's getting colder for longer over the winter, and the trees were flowering, or the crop was flowering, but there was nothing to pollinate it. So they were seeing yield decreases just because these two organisms that have co-evolved are now slightly mismatched. And I think that's a product of climate change that a lot of people wouldn't be aware of. The idea that there's all these systems that have synced up over thousands, millions of years, and just because of slight changes in temperature, we see these mismatches, and it's much more of a problem than I think people recognise. This might not be an appropriate question, considering this was your master's, and you have to sort of do what you have to do at master's level. It's a lot less self-driven. But was there a specific knowledge gap that you were trying to address, or that maybe your supervisor was trying to get you to address?
No, I mean, I think that certainly my supervisor had ideas of where he wanted the paper to sit. It was because his research group is plant micro interaction, stuff like this. So he was particularly interested, I think, as to how change in phonology could affect these interactions between pathogens, in this case powdery mildew, any herbivory, stuff like this. I think he wanted it to fill that gap. Yes, I certainly at the time was happy to go along with the sort of journal idea. But yes, then you had to sort of make it your own study by the end, and put the work in.
Yeah. Were there any specific questions that you were trying to address?
So with this one, it would be how does germination timing affect plant performance and survival based on any interactions with light availability, herbivore and pathogen attack. Right.
And did you have any idea what you were going to find at all? Did you have sort of
so it's difficult, this, because part of the reason we did this study was making it a little bit noisy. So we were putting it into the field, because you can do lots of sort of very more lab-based, add just one herbivore, add just one pathogen, see what happens when we sort of move the variables. But the issue with that is you don't get an idea of how it works in the whole system. So with this one, we wanted to put it out into the field to partly see what would happen. So we had ideas. We had, so stuff like, high light availability might decrease powdery mildew levels. Maybe early phonology, early germinating seedlings would have decreased herbivory by the end, or something like this. But I mean, these were sort of, you could find lots of papers that disagreed and agreed with it anyway. So you sort of had to do it and find out, and then sort of pick the interesting things from there, I think.
Absolutely. So you've set the scene very well. I get a real understanding, I think, why you did this, which is not always the case with master's students. I know that if you read my master's dissertation,
it didn't make the journals I read, unfortunately. I was
it was not great. It was okay. I've got it on my laptop and I cannot look at it. I've tried to look.
No, this
quite
is painful looking over this.
That is. I think if it's not, there's a problem. Like if you like, oh yeah, this is so good.
No.
There's something wrong with you. You need to have that little bit of self-doubt. Self-hate. So let's dive in now into what you actually did. So we're cracking on with the materials and the methods section, which I think is always my favourite section, because it gives you an understanding of a person doing some work. So you've told us when, but where was this study conducted?
So this was conducted in a field around the back of the Burgess Botanical Gardens and the Natural History Museum. It was a little bit of land that, because I was told by my supervisor, it was like, go find somewhere to do the study. And that was sort of it. And everyone spoke Swedish. I was like, right, okay. So
this was in Sweden.
Yeah. So I had to
That would be weird if it wasn't.
Yes, no.
It was in like London.
London, everyone's speaking Swedish. But
Barnes. Lots of Swedish people in Barnes.
Indeed. But so it was part of, at my master's at Stockholm. So I ended up doing it in this field because I'd sort of been wandering around the university areas and I'd found somewhere that I could set it up. And so what I did is I set 3 different blocks of distance from the nearest forest line, just in case there was any effect of, like, we were calling this basically the source of powdery mildew. And initially I had to sort of germinate all the acorns. There's normally not great success rate with acorn germination. So, I mean, there was a lot of acorns, like thousands and thousands and thousands, just to sort of get a good replicate number that you then plant out. It was a mixture of acorns that I'd collected some just from around Stockholm, which was, very pleasant to wander around and see everywhere, and you find some maternal trees and collect acorns. And then just to keep the sort of replicate numbers good enough, I also had to get some delivered from the Netherlands and stuff like this. So I set it all out in these blocks. It was a particularly painful experience setting up the experiment because it very quickly gets out of hand. So you're like, great, I'll set up an experiment, these amount of trees, saplings, etc., etc. Then you realise, I've got to dig each one into the ground because otherwise they'll die of, the sun and dehydration. And then you're like, oh, right, but I also need to set up an electric fence around the site that's at least 6 feet tall because otherwise the deer will just eat everything. Well, at least they could eat everything. So you end up learning how electric fences work. You build one yourself and stuff like this. I'd end up in the middle of nowhere in Sweden. Just it was like this industrial estate, going around and around hangars and trying to find the place where I was going to buy my electric fence from. Then loading it into. I didn't have a car at the time either, so I had to go get it on public transport. So I've got this massive box of like all my wire and poles. And just the look on their faces when they gave it to me, and they were like, "Why? What are you doing?" But no. And then so then I'd set it up. I dug all the holes, which was painful. Especially it was like the hottest summer in Sweden for 206 years, something like this.
Wow.
I'd also taken up rugby at the time.
Oh, really?
So I was
Sweden.
Yeah.
They play rugby in Sweden?
Yeah, jag vill spela rugby. So it was quite a small club. I'm going to detour to talk about my club now.
That's all right.
It was an inclusive rugby club.
Cool.
So it was really good fun. It was the Stockholm Berserkers. It was a smallish team sort of growing. At the time I didn't realise it was an inclusive rugby club. I just turned up and I was like, oh yeah, I'd like to play some rugby. And then suddenly you realise like, oh, everyone's actually quite nice. There's no dicks anywhere like this, Which
is unusual.
Yeah. Well, rugby clubs are usually a bit like, oh yeah, this guy, oh yeah, sure. But no, everyone was lovely, and it was just a great experience. But it meant I was doing trainings Tuesday and Friday, and then anytime I wasn't doing rugby training, I was digging holes. And
you must have been fit.
I was. Yeah, it was fit's a word. I was definitely very fit, but also damaged.
Run down. Maybe.
Yeah.
Overtrained.
I'd like broken 2 ribs on a match, and then realised I had to still go out and like set up my field site afterwards. So it really was this sort of you're there, you can't really dig so well, but you still need to get. It was painful. But
I understand completely. I did a season of sampling for a company. Back home. So I would have to go out and dig rows of potatoes up, and I was playing rugby on the weekends and just getting broken.
Yes.
And then I would be there with my spade or my fork. It was brutal. What position did you play?
I was scrum half.
Scrum half.
After my stature. I wasn't
Not a tall man.
No, not a tall man, not a big man.
Especially not compared to the Swedes.
No. But as the fortunate thing was the level I was playing, it was generally amateur. Mostly amateur. There was, for some reason, we had the Uppsala we played against. Half of their team were like the professional team for Sweden. And I'd lost so much weight doing the rugby and this, I was down to something like 65 kilograms.
Wow.
Just being like pushed around on the ball and stuff like this. It was like, yeah.
Bit of agility though, get around them.
Yeah, that was the aim, you know, which certainly, more or less worked. The season I played, we performed better than we did last year, I think, but we still received like a spanking of 100 to 5 from Uppsala.
That's a tough day.
Yeah,
a really tough day. Been on the end of those.
Yeah.
Well, that's great. It's always nice to hear a little bit more about the person you're talking to than just the hard facts.
Yes.
And you've answered almost everything I've got written down here. It was a great summary of your methods.
Nice.
One thing that I think you did leave out is what you were trying to manipulate. What was your independent variable, to use the scientific term?
I had 2 levels of light availability. So this was just shade netting that I put over the top. So I was doing that in my blocks. The other manipulation obviously was the Acorn germination. So I'd gone in the greenhouses before. And they were kept in cold storage, and then when I wanted to create a phonological block, I'd take them out and try and germinate them for a specific time. So it meant I had 3 levels of germination, sort of early, intermediate and late, and then I had 2 levels of light availability, which sort of high and low. This was, you're more or less trying to half replicate sort of a particularly closed off canopy or sort of more open canopy, etc. I would have liked also a sort of completely open canopy, but as I mentioned, hottest summer for 206 years, anything in that heat would have just died without any shade. So that would have been a fairly obvious result. So do that. But yeah.
How difficult is it to get your acorns to germinate? Is it as frustrating as getting potatoes to sprout?
Yeah, it's potatoes hard, are they? I'm like,
Can be. I think I just don't know the right way to do it. Because all the conventional wisdom says put them in the light.
Yeah.
So I've been doing that, and they sprout quickly. But then I think that light, that getting used to the light, then stunts them slightly when you put them in the dark, when you bury them.
Fair.
So I've been experimenting a little bit, and I think trying to germinate them in the heat but the dark would be a better idea.
Fair. I think the actual germination isn't too bad as long as they haven't died, and I think that part of the issue is because they're recalcitrant, meaning they'll dry out, and once they've dried out they won't germinate. So it's hard to tell whether the acorn you have will be viable. So this is the main issue I think you find. In terms of actual then germinating, I mean, as long as they've been in cold storage for a little bit, plop them in and then they should go well. But yeah, you'll plant 100 and then you'll realise that, 50 of them must have already been dead or something like this. Some of my bags I'd collected, I think the germination rate was like as low as 2% or something this. Yeah.
Presumably you had to plan for that. Did anyone warn you that was going to be the case?
Yeah, so the way I was germinating these was I was getting massive trays filled with soil, and then you can sort of place them all out. So rather than doing it in one individual plot, hoping it grows, then you can see which ones are germinating and then take them and then pot them up and stuff like this.
Yeah.
And then as soon as they germinated, we moved them outside because we wanted them to be affected by the whole climate at the time and stuff like this.
Yeah, but would they maybe not have germinated had you planted them outside?
Possibly not. It's mostly as you wouldn't have been able to tell until it was a bit too late because you've had to put it in a pot and then go around and be like, well, those ones didn't germinate, and empty them. Also, yeah, you're a bit worried that at that point, before it's formed into anything, it could be eaten by a herbivore. Squirrels always, I think anybody that works with oaks generally hates squirrels with a passion. They're always talking about how much, they hate them and they damage the trees, they eat the acorns, all this stuff. But so basically, there was this sort of pre-germination in the labs, which helped a lot more to get.
Because that's something that I can see a reviewer getting a bee in their bonnet about. Trying to be naturalistic by doing it in the field, but
it was
because
that was a reviewer comment. So I think somewhere in there's a sentence that basically says it's fine, mate. It's like we were like, because they were like, well, you're doing it in the lab, but then moving it out, and we were basically saying, well, it's still, it's being moved out as soon as it germinates. So it's not like we're growing them in the lab for a certain height or anything like this. It's just to sort of instigate that germination. And then they're still out for the whole sort of natural range of what oaks would be.
And if you don't do it that way, you might not have an experiment.
Yes,
which is something that people forget, I think.
Yes.
It's like, yes, we did have to do it that way. Yeah, I think it's important to acknowledge that it's not, ideal, but otherwise, how are we going to collect all this data?
Exactly.
Is there much variability? Obviously, you've said there's a very low rate of germination in the acorns. Is there much variability in how long it takes them to germinate? Do you remember?
Yeah, so it, again, some will, it's what made it difficult again, where, some will germinate after X amount of weeks. And I think there's some level as to, because they need to be in cold storage first to sort of go dormant, and then they can germinate. So I think it's just a level of where some maybe they're in the centre of the bag, or it was a bit warmer or something like this, so they didn't really have enough cold storage. Even I've been just germinating acorns at Reading, just from trees I collected at Whiteham. And I did my first round of germinations, and I did proper germination tests to see what would happen, and then I got the results from that, and then all the ones that didn't germinate I just chucked back in the bags and back in the fridge. But then, based off that, checking in my fridge, acorns everywhere, germinating, they're still trying and stuff like this. So it's,
I have spuds sprouting in the fridge at the moment as well. They're just not a lot you can do to stop them.
No, but in terms of what I was actually recording after that, though, because I was trying to get an idea of how powdery mildew and herbivory attack affected these seedlings. So I was doing general performance traits in terms of measuring how tall the seedling would grow, how many leaves, leaf width, stuff like this. And I had to individually label each leaf. So I had I had 700 odd saplings. I was hoping each sapling would produce about 3 to 4 leaves, something like this. Then there was just instead this huge amount of bifurcation. Anyway, but basically lots of branching. So the average was probably closer to about 8 or 9, with some little saplings having like 20 odd leaves and stuff like this. So I'm there with my little Sharpie, sort of bent over under the shade nets, marking each one
with your broken ribs.
Exactly. Yeah, and then you've got to mark each one, and then go back and record powdery mildew on the top and the bottom, herbivore attack, chlorophyll, leaf thickness, seedling height, and all this stuff.
I have an experiment planned to look at leaf greenness in potato, and I just finished I'm finishing an experiment where I did it, and I've decided that I need to track the leaves so that I can try and stay on one leaf, because it's not something that people normally do, but I am slightly dreading that, and that's only going to be for a few plants.
Yes.
But you had to count every and record every leaf on every sapling.
Arguably, it was probably I probably shouldn't have done as much, but you have the plan, the protocol, and you're like, well, I'll follow this, and then when it gets out of hand, you're still just trying to do it as fast as possible. I probably could have gotten away with not recording every leaf on the plant or something like this, but I did. So
you got the data, so why not? I think that is a good bit of advice for when you're designing an experiment, is try and collect as much data as you can. But be aware that you may have given yourself too much work.
Yes. And there becomes a level when it's like, well, this is now too much. And is this actually getting me much more?
And you want to prioritise what you're collecting. There are more important things than knowing exactly how many leaves everything's got. Especially when you're looking at predation and parasitism and pathogens. You have already mentioned that you had to buy and learn how to put up an electric fence.
Yes.
And I wondered why you chose to exclude large herbivores like deer.
I this was another comment from one of the reviewers. I think this one didn't like
I should be a reviewer.
And it's one of those things again where we just had to say, well, the danger was, because this was a 1-year project, I was in Stockholm for the year, I couldn't come back after this, so we wanted to make sure we still had something to record. So in the off chance that a single deer came along and just ate everything, we sort of had to exclude them. And I don't think it's incorrect to do that, because we're basically saying, well, another study can find out the effects of deer on our seedling, sapling, stuff like this. We're specifically looking at those that haven't been attacked by large herbivores, whether there's an effect on powdery mildew, small mammal herbivory, just general insect herbivory, stuff like this.
Yeah, I guess the argument that a reviewer might make, and it's sort of extremely pedantic, but they might say that there might be an interaction between large herbivores and small herbivores, and that may alter the relationships that you see, but what can you do?
Why don't you do the study then, Dom?
I would be terrible. I would be absolutely terrible. If I had a 0.2% germination rate, I would lose my head. And sometimes you just have to make these decisions, and
absolutely.
Research can't be perfect, but you've still got to try and learn as much as you can. And who knows if there is even a relationship between big deer and there are in all the Disney films, but maybe in real life they they do their own thing. Great. So I think we've got a good idea of what you've got up to and how you did this study, and you've answered all of my questions without me having to ask almost any of them, which is really good stuff. So now we can move on to results. Results are the piece of the paper that I struggle to read the most. Because it is very dry. It is literally just... I mean, your graphs are lovely, and I'll show the people at home.
I've gone for they're supposed to be colourblind friendly. That was why I chose those colours.
I try and use greens and blues, and avoid reds. Very good, very good. And these are strong graphs. Tables, I can never get on board with a table. Can't stand tables. So to try and make this bearable for me, could you just highlight the most important or interesting results, or are there any results that you found unexpected?
So the most unexpected and possibly, it was the small mammal herbivory. So we weren't really planning as such to record small mammal herbivory. It catches in that small mammal herbivory. Anyway, we weren't planning to record that as such, but then it came about that, that stuff started getting eaten, which we weren't quite expecting. And then we were like, well, we'll record which ones are being eaten, because it's still an effect on plant survival, plant performance then, obviously. And what we found was this very strong effect of the early germinating seedlings basically had very low mortality rate. So they mostly survived. They weren't really eaten. I think it was something like maybe 1%, something like this. But then the late germinating seedlings, they had like 25% of them were eaten. And it felt very much like they were really going after these later ones rather than these early ones. That was quite striking. And sort of an idea we had as to why that might have been was just looking at when the acorns were growing. Those that were earlier, they had more chance to sort of grow tall and large, and I think most importantly the acorn they were growing from shriveled quite a lot, because obviously all the stored carbohydrates are coming out from there in to make this seedling. All the late ones, because they started growing so late, they weren't that tall, and the acorn was still the sort of this large juicy thing. So it seemed like basically these mice and voles and stuff were wandering around, seeing the ones that were looking the tastiest, and then sort of once they'd ate that, it was something 99% of the time, once they'd ate the acorn, the seedling wouldn't grow and would die. Very occasionally they'd eat the whole acorn, but sort of leave it more or less attached to its roots. And then it would still grow, but it would then, because of all these stored carbohydrates you'd have in the acorn, they couldn't have that anymore. It was these tiny, piddly little things and stuff like this. So that was the most surprising and really interesting, because then you're saying, because there's these ideas where maybe you want to germinate earlier, so you've got a longer growing time, but the issue of germinating earlier is maybe you're going to get attacked by frost or something like this and impact your survival. But we were saying, well, even if you germinate early, it seems like you might have a reduced impact of being attacked by voles and mice. So maybe it still was a good idea to sort of germinate early. In terms of effects on powdery mildew and whether there was interaction with light availability, I think there were a host of different things where it seemed like there was occasionally stuff where, maybe under higher light there was a bit more powdery mildew. But what we found, because we did the overwinter survival of this as well, so after I'd done my study, I'd gone back the next year because I was still working as a field assistant, and then seen which one survived to the next spring. And there wasn't really any effect of powdery mildew or different light treatments or insect herbivory there. So we we seem to basically be saying it wasn't so important, the sort of environment, but the phonology did seem to be quite important, whether that was, just you're more likely to get eaten and die versus, anything else. So, yeah.
Yeah, and that brings up quite nicely the idea that I think people don't really think about, but this is so important in biology, is this idea of trade-offs. There presumably, from this research, there is a trade-off between germinating late and avoiding the frost, and germinating early and being eaten by animals.
Hello.
A man has just come in to clean the door handle. This happens in every single one I do, and I don't know what they're doing. I don't know why they're doing this.
It doesn't seem important to do it now, does it?
It doesn't seem important to do it ever. To be honest,
it's like he's got a microphone.
Yeah.
Tell you what.
I don't know why they, and nothing else is rubbed down. It's just the door handles. And I'd seen this actually. So I had COVID in January, and I'd been in the library and I'd seen this guy. It's always the same guy going around cleaning the door handles. Nice. So I had it in my head, and then got COVID. And they ring you up like a million times a day just to make sure you're not doing anything and to make sure you're sane, because it is difficult when you're isolated with your girlfriend for 10 days. Sorry, Ellie. And I said, do I need to clean the door handles? Because I've seen people at uni doing it, and they're like, no.
What, you're licking door handles? What are you doing?
They literally were unless you're spitting on it or licking it, or why would you need to?
Got to get that grip.
Yeah, crazy. But they still do it here for some reason. I think he's just bored. Because he's always coming around doing it. No matter what time I'm in here.
Maybe he just wants to chat. Maybe he wants to be a guest.
Maybe I should ask him if he's got any recently published work to talk about. Maybe he could do a study on cleaning door handles. He could leave half. That would be unethical though.
Okay.
You get in trouble with that. Great. So you weren't expecting to see this small herbivore predation effect, but you noticed it early on, and you thought, we'll just collect this data.
Yeah, more or less. We noticed it mostly because I was worried all of my saplings were going to die. And I was like, oh, okay then. And then at that point you're like, well, there's nothing I can do because, at that point I'd done so much to set up the field site. You could start talking about, we'll get chicken wire in to try and build, but it was right in the peak of when I was supposed to be recording stuff anyway, so you just don't have the time. So you've just got to, again, accept it and be like, well, we'll see what happens then. And luckily there was instead this just sort of, really nice pattern that then you can sort of talk about and, maybe says interesting things. But
definitely, the more data you collect, the luckier you're going to get. Because you're going to have access to more things you can look at.
No, indeed.
So well done for noting that as a master's student. Because I would not have done that. Great. And do you think? I mean, obviously, this small mammal, presumably small herbivore predation, was unexpected. But going back to what you said about the questions you had before you started, do you feel like the method that you employed and the study that you did answered those questions well? Do you think you could have done it differently?
I think the method was reasonably good. Ideally, I think you want to follow them for a couple of years afterwards to see, because there could have been this thing where there were effects of powdery mildew or herbivory, but maybe they weren't going to be noticed until later on. But you don't have the time, obviously, to carry on recording. So yeah, I'd obviously have liked to record it for longer, a few seasons, to see if there was any effect there. Obviously, I'd have liked maybe greater variety in the shade netting. But, with the heat wave, basically anything that wasn't under shade netting was going to die. So we didn't have a lot of option there either. But I mean, I think the methods generally, I think, were fairly good. I think it is just this element of you have to slightly see what you get. And I don't know if there's anything explicitly that I'd change for it for this study, because I think it was still fairly good at finding out if there was an effect of spring phonology, sort of germination, with any later on effect of powdery mildew and herbivore attack.
Did you manage to collect any data on the herbivores that were predating your seedlings?
No. So yes, I suppose that would be something you'd like to do as well. But this is part of the reason you can know which damage there is, whether it be sort of chewing or piercing and stuff like this. But without sort of really experimentally increase or decrease in herbivores, you're not quite going to know what's attacking your seedlings. So this again is why we sort of done it in the field was, we don't know quite what's going to attack it, but we can just talk about generally the levels of herbivore damage rather than specifically which ones.
Would have been nice to have, camera traps or something. Cost money. And
I mean, the person...
More effort than they're worth, I think, a lot of times.
There was a PhD student after me who worked on my field site, and one of her projects, she attached aphids onto the leaves. It was basically, she was seeing...
How did she do that?
Well, here's the thing. So she was seeing if, I think it was group size, whether there was effect on group size in this really small scale. And literally she glue little strings to their backs and then attached to the leaves, so they had this sort of roaming area they could go around
like a dog on a lead.
Exactly. That
must have been frustrating.
Well, as soon as there's these tiny things, I think that you can do it, but oh god, so much pain. Plus I don't think she even then, because that was later published, but I don't think she ended up talking about that, particularly the aphids. She ended up just talking about how early phonology affects later senescence, basically.
I'd be talking about it if I'd done that. If I'd been through that hell, I'd be like, we're talking about this. Nothing interesting came of it.
Exactly.
But I want it to be published that I did this. And it took forever. So this is maybe a question you've already answered. But do you know? You might not know this, but do you think at least that wild acorn germination coincides with the population of acorns that had the highest survivability in your study? So that early, late, intermediate germination. Because you'd think it.
I mean, if I was asked to bet, I'd probably go with the results of my study. Yes, I understand there's lots of different factors going on there, because obviously you've got not just got germination, you've got sort of predispersal acorn predation. You've then got where it's landing in a specific area. Like all of our acorns were potted to sort of stop any effects of the ground or anything like this. There wasn't any competing plants, because that was another thing where put down ground sheeting, so we didn't have to worry about that. So I mean, educated guess, sure, I'll go for my study getting it right, but you're not going to know, and then until you sort of do these field studies and then see if there's any relation there.
I've written a question. I don't fully understand what I mean by it, so I think I might skip that one.
Okay.
I'm not really sure what that means.
Second glass of wine, was it, when you were writing?
Perhaps, yes. Something that I did find very interesting, you mentioned briefly towards the end of your discussion, and this sort of tickled my fancy because I have a interest in evolution. But you suggested that the deeply buried early germinating seedlings, not necessarily in your study but in the wild, might have a lower mortality than more shallowly buried later germinating seedlings. And you said that this may have something to do with squirrels, which obviously the bane of your existence when you're trying to do the research, but in a natural environment, very important part of that. And I wondered if that was possibly an example of a sort of coevolution, maybe with the oak trees and the squirrels. Are they do they piggyback on the squirrels burying them to get that earlier germination? And also, why do you think deeper buried seeds would germinate more quickly?
So I think specifically with the depth of the seeds, that was more talking about because I think it was a reviewer comment had said this, and it was because we've potted them up, the soil's a bit loose, and so the ones that sort of were getting eaten, they were sort of could be dug into a bit easier. So then we were talking just generally about where it might be the ones that were buried particularly deep in the wild, and then sort of forgotten about by a forgetful squirrel. Yes, they'd probably do better because maybe those that aren't buried so deep can still be eaten by another seed predator, something like this. And there doesn't really seem to be any issue with an acorn growing from particularly deep. I think this was partly just an allowance we'd made in our study because pots, it was actually fairly easy for the seed predator to get into. Yes, it was more or less. Well, I think have I answered that? I'm not too sure.
I think you've answered it pretty well. It was a very long question. I maybe should have broken it up slightly.
No.
But yeah, just the idea that, the oak trees might be taking advantage of squirrels in some ways. They might have to sacrifice a certain, as I guess is the case with masting, which we're looking at now. It's a sort of sacrifice of big yield to keep the carrying capacity low.
You're sort of sacrificing your acorns, but I think most importantly, you're sacrificing your neighbour's acorns because you're trying to do it all at the same time so that, yes, your individual acorn has an increased chance of making it. And there is definitely studies that talk about the distance that the acorn is taken from the original tree and the sort of depths it's buried at, and stuff like this. But
great. So overall, do you think, and this is a really difficult question to answer, but do you think your study was successful?
I mean, I got a master's. That was pretty good.
Yeah. It was published. It's more than most people achieve in their masters.
No. So, I mean, it's difficult. I don't know if you can talk about papers as being successful or unsuccessful. I suppose it's unsuccessful if you think there are things in your study that have, been biased or there's some sort of artifact that you're not allowing for. I think in terms of what this study was trying to answer, it's done the right thing to sort of generate an answer there. It's not necessarily as easy as a yes or no answer all the time, but the study was looking at these sort of interactions and whether there was effective phonology and light availability, and it produced what it produced. So I don't know whether it's right or wrong per se, but to the best of our knowledge, it was successful in that.
That's a very, very good answer. Very scientific answer. Did the paper address what we set out for it to address? Were our methods appropriate and valid? Very good, very good.
You find that said more when you don't find something really interesting.
It's a great success. We learn all this cool stuff.
Science.
I know what dark matter is now. So what's next? I mean, obviously, this is your master's. You're now doing your PhD. What are you planning to do next big experiment?
So, yes, it's not going to be related to this paper, next big experiment. As I said, there was stuff that came from this. So, a visiting PhD student was able to use my field site, and she published her paper that used a bit of that.
That's a really good graph.
Thank you.
Really good. Sorry.
No, it's good. It took me ages as well. So,
very good.
Just a thing, if everybody ever writes in data, never write it in columns, even though it makes sense to you. Always write it as sort of melted. So I'm not going to be able to explain myself well here, but so with these, I had early, medium, and late phonology and stuff like this, and then different light availabilities. So I think I had the tree 1 here, for instance, and then I'd listed whether it was early phonology, what the light availability was, etc., etc. The issue with that is when I went to then make the graphs, they weren't, the graphs the program couldn't understand how it worked. So in the end, I had to, you take them all and you put them all in one column, and then you just have a label at the top saying tell me what stage or light availability this is in, and then you just put it in, and then that seemed to work quite well. I don't know if any of that made sense, but
not to me.
It was a great bane of my existence.
Yeah, not particularly data minded, so maybe to someone it's made sense.
But yeah, so in terms of what's next for this, this one is pretty much done. So instead, I am doing my studies on oak masting. I'm doing a study into individual variation of acorn production at White and Woods. This is looking at, because mast years, you get this idea that a huge area produces acorns at the same time. It's not actually true as such. You still get some trees producing the vast majority of the acorn crop and others producing basically none. So I think this is fairly interesting to sort of research to find out whether there's a particular reason why these ones are producing more, whether it's, maybe they're just taking the mechanistic drivers are affecting that one more, and then can you work out what the drivers are, because this one's doing really well and this one isn't doing really well. Is there a difference in the environment there perhaps? And from that, I also want to do some sort of intervention studies. It might be looking at pollen limitation or something like this, whether self-pollination, outcrossing pollination on oak flowers produces more or less acorns of better or worse quality or something like this.
It's an absolutely fascinating thing to research, because there's so much going on, and there's so much that could be interacting with the trees, because you've obviously got the environment, you've got the more general climate, you've got like pollen issues, pollinators, predators, so much.
There's always a lot to look at. I think part of the issue, because there's a lot to look at, every study you then get this. It's similar to you pointing out the large herbivores earlier, where you're studying something that's very, very complex. You can only study X amount of it. So then you always have someone who comes and goes like, well, why didn't you put this in as well? I'm like, well, there was only so much.
Do you want to pay for it?
Yeah.
Do you want to come and help me collect the data?
No,
Science has to be reductionist. It just has to be.
I'm trying to fight against that somewhat with my sort of first study, because I'm trying to look at everything, if possible. Or look at the main areas. So looking at pollen limitation, any sort of nutrients, and just general environment and microclimate. So hopefully I'll cover my bases there.
Lovely stuff. I think that's covered pretty much everything I wanted to ask you about. Absolutely fascinating work. Is there anything I've missed that you would like to talk about?
I don't think so. I think, we covered this generally. I apologise, my memory of it's not that great. I'm sure I'll have a very good job. Said stuff that's incorrect or just made stuff up, I'm sure. But,
if people want to read it, it is, once again, Spring Phonology dominates over light availability in affecting seedling performance and plant attack during the growing season, published in Forest Ecology and Management. Is there anyone you want to, mention, thank, any of that sort of stuff?
Well, I mean, I think the paper, in terms of obviously all my co-authors there, these were the ones that, guided me to write a paper and generally helped me throughout my whole master's experience. They were a good group of people, especially when you are at that level where you don't really know anything at the beginning. You get somewhere.
So you mean like right now?
Well, exactly. You do your whole undergrad, and I think it's very easy for you to do your undergrad with actually, huge gaps of knowledge in certain areas.
Absolutely.
So I think the main one for this was the sort of stats knowledge. I didn't know how statistics worked.
I still don't. No.
Yeah. Well, nobody knows how they work.
No, that's true.
I definitely didn't know how
Statisticians claim they know.
Well, exactly.
But it could just be magic. Honestly, it could just be magic.
They were there. And then I would also, because often with my work, my girlfriend is always there helping me collect data. And she never gets her name on anything other than the acknowledgements.
Is she in the acknowledgements?
She's in the acknowledgements. She's the only one I thank right at the end.
We thank Eleanor Child for help with setting up the experiment and recording data. So thank you, Eleanor Child.
Indeed.
My girlfriend's called Eleanor as well.
Oh, yeah. Not Eleanor Child.
No, not Eleanor Child. No.
Awkward, wouldn't it?
No, and if it was, I wouldn't tell you. No, Eleanor Froud. Odd name. Even she misspells it sometimes. Her name on Snapchat when we were at school was Eleanor Fruce. Because she put an S instead of a D on her own name. But I wouldn't let her anywhere near my plants. But no, that's great that you go out and collect data as a team. That's very, very helpful of her. A rare thing.
No, I definitely wouldn't have done as well as I did in the masters without her, because it's even little things in terms of so I'm there bent over collecting data, trying to count the powdery mildew spores on my leaf. And if you have to do that with a clipboard to write it down, you can't put so you just need someone there to sort of be acting as scribe for you and stuff like that.
Yeah, it's so helpful.
Someone also to be like, say it's 2:00 in the afternoon, you're fed up and tired and you just want to go home. So I'll be like, let's just do a few more, because someone will have to be here the other day.
I feel like I need that, but not from my missus. She was great actually when I did do my masters, because she paid slightly more of the rent. Because I was very, very poor. But
emotional support, any of this though, surely.
A bit.
Oh yeah.
Her emotional support generally is just like, suck it up.
All right.
That's what you're doing. Come on, mate. Stop moaning. Because I moan a lot. I am a moaner. So she's like, stop moaning.
Fair.
I should stop moaning. But this was great. This was a really good episode. I think you've done an exceptional job of explaining what you did, considering it was 3-ish years ago. And thank you very much for coming and doing this.
Thank you for having me.
Hopefully you'll get some more stuff published during your PhD and we can chat again.
Hopefully so.
And thank you very much for watching and listening. I always do that. I point at the microphone when I say listening, which they can't see. But thank you for watching and thank you for listening. I don't need to point. And I will see you in the next episode, although I don't know who that's going to be with or when it's going to be. So if you know anyone that's published anything in your
I mean, there
sphere.
must be people, surely.
There's always people. It's just getting hold of them and getting them to agree to it that's difficult. It's a little bit easier at the moment.
It's all that. It's just that research self-hate there. It's like, oh no, I don't want to talk about it.
Yeah, but it's fine. It's fine. We all have that, so we can all relate, and and also it's nice. I think it's nice because normally, you go to the pub or whatever with friends from home or school or whatever, and you're like, let me talk about oak masting or potatoes. And they go, no, shut up. We're here to watch the rugby or whatever it is. So, I think the people that have done it have enjoyed the opportunity to talk about their research without, the normal experience of people going, shut up, please. But thank you very much. This has been great. And I will see you or you will hear me in the next one. And I always do this.
It's a strong ending.
This is embarrassing, but I always do this. I have to do this because there's a guy who specifically requests it. Peace.
Nice.